Professor Kathleen Collins from the Department of Molecular and Cell Biology here on campus. She's been a member of the faculty here since 1995. After undergraduate studies at Yale and graduate school at MIT, she did a postdoctoral fellowship with Nobel Prize winner Carol Greider at Cold Spring Harbor. She's received a number of awards, including American Society of Cell Biology, Promega Early Career Life Scientist Award, Burroughs Wellcome Fund New Investigator Award in the Pharmacological Sciences, and I hear her interests, aside from her research, include growing strawberries and relaxing in the Berkeley YMCA pool, something that I'm sure some of you also enjoy. But Professor Collins, thanks very much for speaking to us this morning about aging.
Is this good volume in the back?
Yeah.
Okay, good, great. So thanks, Steve. Thanks for coming and listening. It's a great pleasure to have people actually interested in what you might want to say on a Saturday at 11 in the morning. So today I'll tell you about aging versus immortality, mostly at the cellular level. In the Collins lab here on campus, we have many thoughts about aging and immortality, and we draw them out on our lab wall. So this is just a page rendition of our faculty views. It's in one of our lab rooms, and so we try to think about how to stay young. The story today will take you from the molecular level of DNA strands to the DNA strands packaged into a cell, the cellular level. So this is an example here of human cells that are in each one of these culture dishes with media that's red. And then the story will go from this cellular level to multicellular organisms in these three stages. So the progression of today's story will first be about replicating the genetic code, and in particular about some special needs DNA ends. Then cell proliferation, which means the expansion of cell number. And also how this proliferation in multicellular organisms is coupled to making those cells take identity. And then we'll get to the organism level, and we'll talk about aging as a countdown clock, and how this countdown clock plays out biologically versus immortality. And because this is Science at Cal, at the end of each one of these sections, I'm going to show you an actual experiment. So you can ask yourself, how was this done? And it will tell you about Science at Cal. So to begin with the DNA. The DNA genetic code, as you most likely all know, has 4 bases. And these 4 bases are polymerized as head-to-tail chains. The 4 units, obviously, not that much information, but if you read the chain in groups of 3 or 4 or 5 or 6 pretty rapidly, you get to quite a complexity that can be encoded by these 4 bases. Now those strands, they have a polarity head to tail, 5' to 3' here. And in genomic DNA form here, this single strand is paired to another one, running in the opposite direction. And the pairs have complementary bases. So T always matches to A to T, G to C, and C to G. And these 2 strands then take this familiar right-handed helix, where one strand is here and the other is here. And the base pairs are across the middle. So this is the DNA genetic code that we have, and it's important for today that in order to replicate this code, right, in order to pass this code on, this information content is easy, in fact, to copy, because these strands are complementary. So you can imagine if you take the double-stranded genomic DNA and you split the 2 strands, this one and this one. In fact, this one alone can be used to polymerize the opposite strand because they're complementary, and lo and behold, this one can be used as a template to polymerize the other strand because they're complementary. So to replicate, you split the strands, you restore each single strand to double strand by polymerizing the complementary DNA units. So when I talk about DNA replication or DNA copying, that's what's happening. Now, this DNA genetic code that you have is in very long strands. In fact, here on this slide is your genetic code, all of it. one cell gave up its DNA strands to make this picture. So stained in blue are all of the DNA strands. Each pair of DNA strands is called a chromosome. So one contiguous double-stranded DNA is a chromosome. And then the image below was taken just when those chromosomes had replicated. So here's a double strand, and here's another double strand that's still paired to it because the two parental strands just split and each one got copied. So now there's this chromosome and this chromosome, and very shortly, each of these chromosome pairs will be split between two daughter cells. And the one cell will divide to make two, and each cell will have a copy of the double-stranded genetic code. Now, if you count these, as I'm sure Dan will do, you will see there are 46 chromosomes. And in fact, our code only has 23 chromosomes, so 23 long strands. The reason then that there are 46 chromosomes is because you have a copy of 1 through 23 from mom and a copy of 1 through 23 from dad. So in fact, 46 chromosomes is correct. Now each chromosome here, just for scale, is about 1/1000th the size that you're at the sequence. So they're small, right? And they have long strands, right? But you'll notice here in this slide that one particular sequence that is stained for in yellow here is present at every end. So these 23 chromosomes have different sequence content. But every one of them, in fact, ends with the same sequence that has been marked here in yellow. So this arrangement of linear chromosomes is in fact not the evolutionarily most simple way to transmit your genome. We're comparing chromosome sizes and organizations that vary greatly across biology. So an organism like E. coli, which is a member of the bacteria in this tree of life, it's unicellular. It has one chromosome. And in fact, that chromosome is a circle. So you can imagine that it's pretty easy to replicate a circle. You just keep going around. But in eukaryotes, yeast or fungi, plants, and animals, over here on this part of the tree, our chromosomes are linear. And in fact, the genome gets much larger, and the number of haploid chromosomes, that means from mom or from dad, is, let's say, about 20. Varies with the organism. But this means that, in fact, there are quite a lot of ends. And these strands, then, can be, you can get chromosome 1 from mom separately from chromosome 2. So here, in fact, these genomes are very large, and so most of the DNA in a DNA strand is in the middle. But the ends, in fact, are special, and these eukaryotic linear chromosomes have telomeres and segments that are unique to eukaryotes. So here's a human cell telomere for you. Here we'll blow up this bit right here at the end. And what you see is that at the end, there's a common sequence. It's a simple repeat of 2 T's, an A, and 3 G's repeated a lot of times, running this way, and the complement running this way. Call those telomere repeats. And this bunch of telomere repeats here, present on each end, protects what should be an authentic chromosome end, the telomere, the stable telomere, from the fate that falls to DNA breaks, which are rapidly pasted together, shuffled, and they're kind of damaged. So to not be recognized as random DNA breaks, not to be pasted together, our chromosomes need these telomere caps. So this kind of end protective function requires, in fact, a little single strand overhang here on the 3' end. We call it an overhang of the single strand on the top strand. But in the way that the genetic code is replicated, when you split the strands apart and you make the complement of each to pass them to the daughter cells, you cannot replicate that. So this structure, I've just told you, is absolutely critical, and yet it can't be replicated by copying the other strand. And in fact, one would predict that with each round of chromosome replication, telomeres should get shorter. This is the blue of the chromosome continuing up through the roof, and this is the yellow part that was stained. Each time a bunch of divisions happens, this region containing the telomere should get smaller, smaller, and smaller. And in human cells, this loss amounts to about 100 bases or base pairs per end per cell. So this needs a solution. And there is a solution. And that solution is a special enzyme that is called telomerase, because it makes only telomere. And what telomerase does is come to a telomere end here with this TTAGG 3' overhang, and it makes that overhang longer. It adds more repeats. This breaks the central dogma, because the DNA is supposed to template the DNA. Okay? Only DNA gives rise to more DNA. And here I'm saying this DNA before didn't have these extra repeats, and this enzyme, telomerase, just came and added them. The way that telomerase does this is very interesting. I won't talk in detail about it, but it has a subunit that is both protein and one that is RNA. And the RNA subunit within it contains a sequence that is complementary to this telomere repeat. And so what telomerase does is it uses RNA instead of DNA to template the synthesis of DNA. And this makes it a reverse transcriptase. Transcription goes from DNA to RNA, that's the central dogma flow. This is the opposite. And these kind of enzyme was thought to be unique to viruses. It turns out, in fact, that eukaryotes have it. So now we're at the end of this section, and I'm going to give you science at Cal. This is in 1995. Because this enzyme telomerase was discovered here by Carol Greider as a grad student in Liz Blackburn's lab when they were on this campus. How could you look for this enzyme? How would you have even thought that it could have an RNA template? Where would you look for this? So they looked in this organism here, which is by scale almost big enough for you to see. It's a ciliated protozoa. It's called tetrahymenum. So tetrahymen has a lot of telomeres. And so Carol and Liz were looking in this organism to see how the telomeres might be maintained. And how you do that is you grow up a bunch of cells, you break them open to make what's called an extranode. And what you do is you add a synthetic DNA mimic of this chromosome in it, a single strand of DNA. You also add radio-labeled new DNA units, let that all sit together, and then you ask, did this piece of DNA get longer? And the way you do that is to resolve single-stranded DNA by size. You take a big sheet of very thin jello, you start all the DNA at the top, with a positive electrode down here, and everything runs down here. And the things that are small go fast. So what you're looking at down here would be the size of the unextended DNA primer. And this is an autoradiograph. So what you're looking at here is the fact that that primer got long by 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, by continuous series of this is a telomere repeat, and another, and another, and another, and another, and another. And so this enzyme could make these telomere repeats on this single-stranded DNA without a DNA template. It was carrying its code. So this is how Greider and Blackburn found telomerase, looking in a ciliate for telomere maintenance. And this discovery then, at first, wasn't clear how relevant it was going to be to other organisms. So I'm going to take the progression of today's story, point number 2, and tell you about cell proliferation and cell differentiation in multicellular organisms. The slide of the human cell chromosomes illustrated the fact that telomeres shorten with each cell division. Now in this slide I've been more specific, and I've said telomeres of human somatic cells. So this is an experiment where you take any cell from your body, like a skin cell, you put it in a culture dish, and you let it grow. And as a function of time, you come back and you look for these telomeres. And here, starting at the beginning of that cell division, it's like growth, you can see that telomeres were long, right? But if you let them grow for a while and they proliferate and make more cells, in fact, the telomeres have gotten shorter, and so on, until they stop growing. They stop growing because they don't have enough of that end protective cap to keep the chromosome stable. And the cell says, no more cell division. You don't have enough telomere. So in fact, this came to be termed the telomere mitotic clock. Because up here, the length of telomere you start with, you can grow no matter almost what it is. But every time you duplicate and divide the cell, this countdown clicks off. There's a mitotic clock. Mitosis is the cell division that limits the renewal capacity of these human cells. So hang on a second. I've told you that tetrahymena has telomerase, a mechanism to maintain these telomere repeats. And yes, if you grow tetrahymena in culture forever, it will maintain a certain length of telomere within a range. But cells from multicellular eukaryotes, like humans, like these skin cells right here that are to scale, you put those in a dish, you ask them to divide, and they lose telomere until they halt proliferation. And depending on the cell type, they either kill themselves or they just sit there and eat. So, Liv, how can humans have telomerase if telomeres are always shortening? In fact, it was a challenge to find telomerase in human cells. Because no matter how many purifications of normal cells grown in culture you do, there is no enzyme. So what's the solution? How do we know that human telomerase even exists? The answer to that lies in cancer. Because when these normal somatic cells shorten the telomere, if something like a viral protein expression or mutation lets them continue to have, they have very short telomeres, they go through a replication cycle, and all the chromosomes start fusing. And they're rampant cycles of fusing and breaking. And this rearranges the genome. It's called genomic instability. And in the process, genes get turned on and off. And at very, very low frequency, a cell can survive this. And if it does, and if it activates telomerase, it can become cancer. So a cancer cell will grow out of this, and it will maintain stable telomere lengths because it is aberrantly activated telomerase. And it is thanks to these cancer cells that human telomerase could be identified. In fact, human telomerase was identified in HeLa cells. How many of you have heard about HeLa cells? Okay, great. So these cells were, they are a cell line started in 1951 from a cervical carcinoma, from a cancer. Back in the day, people were trying to grow human cells. You can imagine the frustration. You take a cell from your skin, you put it in a dish, it grows, and then it stops. And the raging controversy was, well, is just the growth media not right, or do human cells just stop? And so it was in the process of continuing to try to grow different kinds of human cells that this line from a woman named Henrietta Lacks, it was abbreviated HeLa, grew. And these individual HeLa cells are shown over here, phase contrast. They're, just for scale again, about 1/10 the size of what you can see by eye. So we have these cells, they're on the bottom of a plate, right here. Here's the tissue culture plate. My student Alex is gonna put these cells in the hood and open this petri dish. But where he got that from was this here. So this is what our incubator in the lab looks like, with the right temperature, carbon dioxide for these human cells to grow. And you can see that there's a bunch of these plates of cells. So these cells grow indefinitely. And that's why we can grow them today when they started growing in 1951. And in part, that's because they have very high telomerase levels and stable telomere length. So if it had not been for HeLa cells, in fact, we would not be able to grow human cells in culture, and we would never have found telomerase. And so these cancer cells, for a very long time, were the only model for human cells, studies of telomeres. But this can't be true, right? Because we don't have to have cancer to have the next generation. So again, there must be some other way to have telomerase on and maintain a human cell telomere. And the answer comes to this point about the fact that we are a multicellular eukaryote. And in multicellular eukaryotes, like us, our body cells, those somatic cells, they don't actually go to the next generation. You can't put your arm in a culture dish and get another human. The somatic cells here, the vast number of cells in your body, they actually don't grow forever. But if you think about it, when a few cells are set aside very early in the development of a human into what's called germline, meaning they will go to the next generation. These germline cells have very few divisions. Then egg and sperm, gametes will come from these germline cells to make a cell that's called a zygotic genome. And this cell will make a bunch of cells, in fact, here in purple, that are the early cells of the embryo. And after proliferating for a while, those early cells of the embryo will decide to make all these somatic cells. But very early in that process, some are set aside to the germline. So in fact, the germline gamete production, here in red, there's only a few cell divisions. And in fact, telomerase is active in some of the early germline cells. Early embryogenesis, okay, here this purple cell, in fact has many, many cell divisions. Because if you think about it, you're starting from one cell, and you have to make an entire organism. Small, big, bigger, and there's a lot of cell turnover. So in fact, it's these cells that would be the ones that should have telomerase. But these cells, human embryonic stem cells, weren't possible to culture in the same way that HeLa cells can be cultured. A cancer has learned how to grow no matter what is around it. But normal cells, well-behaved cells, don't do that. So they're harder to culture. In fact, it was a big challenge for scientists to develop human embryonic stem cells. But my colleague Dirk Hoffmeyer here at Berkeley is a master at them. So here's Science at Cal, number 2 for you. These are human embryonic stem cells, or ES cells, in culture, can be grown. And I'm showing you an experiment here to end this cellular proliferation section. You can take these cells and isolate their genomic DNA. And you take that genomic DNA and you chop it up into smithereens by cutting, for example, at every GATC. Most of the genomic DNA has GATC frequently. In fact, you mix it up and cut with a few things. But the telomeric repeats are constant, T, T, A, G, G, G. These enzymes don't cut at the telomere, so the only thing that's left is telomeric DNA. So then you resolve this DNA by length. Here you leave it base paired, but it's the same. Here's the top, here's the bottom of that gel, and things that are faster are smaller. Here's a scale times 100 base pair length right here. So this is a strand of 2,000 bases. Now you can detect in this resolved DNA the telomeres by taking a little radio-labeled oligonucleotide, this sequence, and you denature all of this DNA, split the strands, and then you let this base pair with the telomere. So everywhere there's a telomeric sequence in the DNA resolved in this gel, you get this signal on an R rate. What you can see is you take wild type, unmodified human embryonic stem cells, and you grow them and grow them and grow them for these days of culture. These telomeres are staying pretty much the same. But if you take telomerase out of those stem cells, no telomerase, now you can see the telomeres shortly, just like a somatic cell. Okay? And eventually, those cells get to short telomeres and die. So embryonic stem cells do, in fact, in human, use telomerase. And it's the embryonic stem cells maintaining their telomeres that solves the problem of our somatic cells not. And I'm going to come back to that. But I want to come to this third point here in the progression of today's story. So I told you that cell proliferation, this expansion of cell number, can happen with telomerase, in which case telomeres are maintained, or not. But in humans, and most multicellular organisms, if you take rapidly proliferating cells, like the embryonic stem cells, and you switch their identity, you tell them to become a somatic cell type, which you can do by changing the ES cell culture media. They differentiate, so they pick an identity, skin or blood, bone, and when they do that, they turn off telomerase. and they're still proliferating, but now they're doing it with shortening telomeres. So this sets up this question. In the sense of aging at the cellular level, in a multicellular organism, how much of the cell division is a countdown clock, that mitotic clock counting down each division? And how much isn't? To what extent do we have cells that are immortal? So I'm going to do, for this, a back of the envelope calculation for you, because in that last slide, we'll say that these telomeres are 10,000 base pairs. Okay? You start off with a stem cell, back of the envelope calculation, and you assume that the human body has 50 trillion cells, and about 50 billion of those are replaced per day. To make 50 trillion cells and replace them, right? How much division would that take? So if you lose 100 base pairs per N per cell division, and you start with 10 kilobase pairs, and you end a short telomere can't get much smaller than 3 kilobase pairs, that's 70 divisions. So if every cell divided this maximum number of times with no cell death, wow, that's a ton of cells. That would be more than enough for long somatic cell life. But each differentiated adult cell type, right, can only be generated from a certain lineage of things called adult stem cells. So the embryo has embryo stem cells. They can become anything. But very early on, you lose those. There aren't cells in your body that can become any kind of cell they want. We have stem cells that are more restricted. They can make, a blood stem cell can make blood, gut stem cell can make gut, skin stem cell can make skin. Okay? So we start off with a fixed number of those. Embryonic stem cells turned into these adult stem cells, and these adult stem cells had a certain telomere length. Okay? So there, ever after that, if you want to make a lot of skin, you're only as good as the telomere length in that adult stem cell. So they're set aside, and furthermore, telomere shortening is often more than 100 base pairs, DNA damage, just random stochastic events. And in fact, in any given cell, there are many telomeres, but it is the shortest telomere that will stop proliferation. So only one of them needs to get short, and then the cell has to stop. So really now, let's reconsider. How relevant is this telomere shortening in the human somatic tissues? And to illustrate this, I am going to use the adult stem cells that come from blood. Okay? The adult stem cell that comes from blood is called a hematopoietic stem cell. There will be a quiz on this later. No, really. Here, this HSC, the point of this is that it's one cell living in the bone marrow makes two types of descendant adult stem cells. So this is an adult stem cell. It further makes two kinds of things that are more restricted. One of them goes on to make a whole bunch of cells that I deleted from the slide, including red blood cells. So only as many, you can only make as many red blood cells as you have bees, right? But on this side, this one here makes a lot of cells also. This is a simplification. The descendants of this one, common lymphoid progenitor, are different types of white blood cells that fight infection, fight different kinds of infection in different ways. And they fight basically anything for it. And so you can see here that this common lymphoid progenitor, it made a B cell lineage that did all kinds of crazy things. It made a T cell lineage that bounced around and became all kinds of different things. So you can imagine that, in fact, you might have a few of these cells, some of these cells, these, and then maybe you use up your long-term memory T cells, right? Well, you can't come over here to this stem cell and ask for T cells. Can't do it. Okay? We can't make more hematopoietic stem cells, and we can't turn this back. So low telomerase activity is present in a few adult stem cells, but in fact, none of them can prevent telomere shortening. Now, fortunately, the hematopoietic system stem cells divide very slowly, because this one only has to divide twice. One of its daughter cells can decide to be this progenitor and make all this stuff, and the other one can stay a stem cell. That's what it means by being a stem cell. So if this divides once a year even, and one of its daughter cells goes to make all this blood for a while, then it can be another year before it comes back and divides again. So these stem cells divide slowly. These ones, there are very few of them, but they divide slowly in a normal environment. But let's say in your circulating blood, you deplete one type by a chronic infection. For example, if HIV gets all your T cells, or EBV gets all your B cells, or you have a deficiency that destabilizes the red blood cells. Now what happens? Okay, the signal got no B cells, okay, goes back to the stem cell and tells it to proliferate more. So through all kinds of events, particularly chronic infection, you can make this cell proliferate chronologically more rapidly, right? If there are more divisions per any given amount of time. And you can, in fact, then think that you might deplete these cells. So, do you really? And now, in order to ask this question, one could take peripheral blood mononuclear cells, basically, we're going to look at these lymphocytes, isolate them from the blood, and ask how long their telomeres are. But instead of doing this with the DNA blot that I showed you before, in fact, there's a way to do this so that each cell is counted differently. It's a flow machine where each cell flows through, and as it flows through, its telomere length is recorded. So each dot on this graph is somebody, a different human, who had their peripheral blood mononuclear cells analyzed for telomere length. And the average of the telomere lengths in their peripheral blood mononuclear cells is shown as a dot. So if you look with chronological human age at just healthy people, you notice two things on this graph. The first is that there is a decline. In fact, that decline is estimated to be very high in the first year of life, continues at a high rate, to about 4 now with 4 data, lightens up a bit, and then declines very slowly over all of adult life.
Is that average or shortest?
This is average. So here, you can imagine that a young child has more cell proliferation to do. The body has to increase in body mass as well as renew itself, right? So there's a higher proliferative demand up here. Also, very young kids are shaping their immune system, and there's a lot of cell killing going on. So it makes complete sense, in fact, that the rate of loss is very high at the beginning and then slows down. So the other thing to notice about this graph is that at any given chronological age, there are a lot of dots. Yes, there's a line through this. But different people have very different average telomere lengths in their blood cells. And this is proven very true. So just because you're 40 doesn't mean you have a certain length. Part of this can be environment, like whether you had a chronic infection, but part of it is also genetic. So telomeres do shorten with age, but there's a wide variance. So is this too much telomere shortening? How would too much telomere shortening manifest in a human? What would it look like? And now we get to Science at Cal. The Science at Cal slide. Now we're in 1999, and we're in my lab. And we were using HeLa cells to study telomerase because we can grow them. And we were identifying what was in that enzyme. And we were kind of scratching our heads because it didn't really make sense. Now, you can't take a human and knock out a gene and ask, well, is there no more telomerase? But what you can do is go to the population and ask, are there people in the population that maybe have a mutation in the gene I'm interested in? Reverse genetics. Instead of making the change you want, you look for the change you want out there. And in fact, there were cells from patients that have a disease called dyskeratosis congenita, or DC, that had been mapped to have a mutation in this protein we thought was important for telomerase. So we asked, well, can that be true? And in fact, if you take Now this is, again, that DNA block where we've run the DNA out by size and we're hybridizing with the telomere fragment. If you take a parent, let's say this was a 45-year-old parent, mother, and you look at the telomeres, that makes sense. This is right here in the middle of this range. That's about right. Age-appropriate telomere length. But this is her 7-year-old son. 7. No, he was older, but teenage, basically. And what you'll see is that these telomeres are very short. They are very short. But they're strikingly short for age. They should be up here. So in fact, we can show that in dyskeratosis congenita, the disease, there is less telomerase at all stages, including in that rail, at about a quarter or a half of its normal level. Okay? So what we learned from this and from subsequent studies by many groups is that if you just half the amount of telomerase, you have bone marrow failure. You take it down to a quarter, you have bone marrow failure in your teens. So in this blot, genomic DNA isolated from the blood cells, chopped it up again, resolved DNA by size, look at the telomeres. These telomeres are so short that these cells shouldn't be proliferating. And in fact, they weren't. And it was really hard to do this experiment because trying to grow these cells was frustrating. And I had grad student Jay Mitchell, and he kept trying to grow enough to make this blot, and it would die. And he would put it in the back burner. But in fact, now we know why. We know why because these cells had such short telomeres, because the people had grown into adults with so low telomerase that they have premature telomere shortening. So since then, in fact, there have been a lot of other mutations that have been linked to this premature telomere shortening in humans. And there are different extents of telomerase loss, and there are different ways in which you inherit the mutation, and many other differences that give a spectrum of defects in phenotype. And all of them, though, are in things like blood cells and in the surface body layers that divide a lot. Skin, nails, hair, lung, gut, all of these things have to renew constantly because they're exposed to the environment. They have very high proliferation rates in adults. So it makes sense, in fact, that if you were going to run out of telomere and certain cells were going to suffer and not be able to divide enough, it would be epithelial lineages in the surface body layers. So I'm just showing here a graph. If you take telomere length in normal people as a function of age, it has a wide variation. In fact, there's a wide difference among people. So you can plot what's here. This is the telomere length for someone, let's say 70 here. This is the 99th percentile. You are very long telomere. Okay, here's the first percentile. Not so good. So if you look at this normal distribution and then you plot the telomere length of people with these diseases, dyskeratosis congenita I told you about, and some others, in fact, it's now a diagnostic criteria for these diseases that the telomere length is short, is below the first percentile for age. And the shorter the telomeres are, the younger the age of disease onset, right? The telomeres become short. Now here, only when this person is 3, they're this short, right? And here, it took them until they were 60 to be that short. So a lot of missing telomerase or little. But there was another thing beyond the level of telomerase that really makes this different. And it makes telomeres different as a type of inherited gene difference. And that's a fact called disease anticipation. So disease anticipation means that in subsequent generations, either the age of onset gets earlier or the symptoms get worse. Okay? So here in increasing shades of purple are earlier onset or worse symptoms in patients. And here's a grandfather, right, who has a mutation that cripples telomerase, but the chromosomes have long telomeres. And remember, there are so few divisions in the germ that this person can get by, make some sperm, pass it to the next generation. But this child inherited both the mutation and shorter starting telomeres, because without enough telomerase, you couldn't reset. So in fact, they have fewer years of somatic cell division before they have disease. And yet again, in the next generation, inherit very short telomeres and the mutation, and this is the most severe one of those diseases on a previous slide. This gives very early onset disease. So you can imagine now what might happen if in this cross, male, female, give rise to son, there had been another child here who hadn't inherited the mutation, but had inherited short telomeres. You got a normal genome, but you inherit from your parent telomeres that are too short. And in fact, studies are beginning to show that those people also get disease, that it takes more than one generation to bring your telomeres back to normal. So it's a very unique sort of way to think about disease.
So besides the rare diseases, they also, I assume, show early signs of premature aging.
These diseases. I'm going to come to normal people in 2 slides. We're now on the aging. Okay. So, yes, these, in this case, these are characterizations of disease. So just one slide in between, point about that. And here I'm just going to show you one slide of mice. So this I took from, obviously this from public magazines, not the scientific literature. The mice are from the scientific literature, they're from the Tapino lab. But if you start with 2 old nieces, if you breed the mice so that mice have actually very long telomeres, very small body size, very short lifespan. But if you rig the mouse such that it's got human-sized telomeres, okay, and no telomerase, it will look like this. This mouse looks a little scruffy. It's missing hair patches. It's got bone marrow failure too. And if you put telomerase back into that mouse's sibling, okay? Now look, with telomerase, the mice are young again. The activation of telomerase in aged nieces rejuvenates entirely their body. Now this is somewhat true. But again, this mouse, like the humans that I talked about with this characteristic genita, they were sensitized by gene disruptions in telomerase. Now out there in the population, there is a great variation in, you're never plus or minus. There's a great variation in how much telomerase you might have. But it's a fair question how these studies, which tell us that short telomeres can limit lifespan, they can limit tissue renewal, how does that relate to telomere length in the general population as a function of age, which I think was your question. And furthermore, in part the jury's out on that, because how much telomere, is this enough over here? But you can imagine that at least in some people, under some circumstances, chronic infection, for example, you do use this up. You use up your telomere reserve. So then it begs the question, why turn off telomerase in the first place if you're going to be able to use it up and have bone marrow?
Is anybody able to measure the shortest length of the shortest chromosome
Sure.
telomer?
Yes.
Why don't you show charts of that?
Because this data is a much larger data set. And my point with this data here is just that there's a lot of variation. So the question is, you already said that it's the shortest telomere that stops the cell from growing. Why are you showing average telomere? And I'm showing average because it's the thing that's more readily measured in the general human population, and average telomere length is a pretty good predictor of the distribution. Okay. So I'm going to come back here now to the end of the story, the progression of today's story. Because this aging countdown clock, we could ask, does it have a function versus an immortality of the somatic cells, if they could continue to grow? And the answer that I have to that is in this graph, which I downloaded from the National Vital Statistics reports from the United States deaths up to 2010. We're looking at the year here, and basically deaths in the human population, in the US, these are US ones, from different causes of death. And the top causes of death are on this slide. And what you can see is that diseases of the heart, they mean heart attacks, have been going down steadily, in fact, along with these cerebrovascular disease, which is stroke. So if you have too much cholesterol in your arteries, you know, you're going to have a problem. But in fact, with the advent of statins and more knowledge, this disease rate is going down. So that's not telomere limited, because actually lifespan is going way up. So you should run out of telomeres more now in a more recent data set. But what you can see here as number 2 cause of death are malignant neoplasms, in other words, cancer. And this rate has not gone down so much. And if we humans are going to die, partly of all kinds of things, Alzheimer's is down here, one major thing we're going to die of is cancer. So perhaps evolution has selected to prevent, to add protections against cancer. And what is commonly cited, and is very true, is that cancer is a disease of aging. There's a sharp increase in cancer incidence with age. You can see that here, age at diagnosis. I actually separated out the male and female cases. You can see that everyone's getting more cancer with age. You take the overall rate, it's here. There's actually a little bit of dip at the very old stages, but very little cancer, and then more and more and more. And this is not linear. This is because in order to become a cancer, in fact, the cell has to accumulate many mutations. So over time, there's more time to accumulate these mutations, and therefore cancer becomes a disease of aging. And in fact, this disease of aging, I would argue, is a good reason to not have telomerase fat in somatic cells. So here's an illustration of how sequential mutations are required to create a tumor. This is a picture here on the top of a normal epithelial cell layer, red and white, and it's sitting on this yellow stripe, which is a basement membrane. So this layer, you can imagine this in your lung or your colon, is doing a very good job of making a barrier, and it's going to absorb nutrients or air across. Those cells know to make one layer. They stop growing when they touch each other. But you can imagine that one of these cells acquires a mutation, and that mutation tells it to divide more than normal. Okay? So in the first mutation, now that red, this little cell right here, is kind of red, it became a lot of cells that were red. But still, that cell is obeying the instruction to grow in a monolith. But you can imagine that a second mutation happens to one of these cells in here, and that second mutation lets them grow on top of each other. Okay? This is honest. This is how cancer happens. And these guys now, they can grow on top of each other, right? But they still can't get out of this epithelial surface. They're either in your lung or in your colon. So in fact, another mutation, you can imagine happening here, that lets them degrade away that basement membrane and sneak into endothelial cells and spread throughout your body. So lots and lots of cell divisions are required to accumulate the mutations, to divide a lot, to become mutants. And in fact, a lot of the mutations are going to cause cell death. And in fact, a lot of these cells are going to be killed by your body because they're behaving inappropriately, and all those white blood cells are going to come eat them. And so they also have to acquire an evasion of cell death. So there's a bunch of hits, multiple hits, that it takes to make a cancer cell. This could sit there for 10 years, become this, become this, become this. And in these cancer cells, though, this unrestricted growth is essential. It's essential to maintain the cancer as that, and it's essential to get there in the first place. So for that reason, you could imagine a balancing act in this telomere length. By turning off telomerase in somatic cells, there's a limitation on this unregulated cell growth. A cancer cell might divide for a while, might become a benign tumor, but if it stops by itself, it's actually not going to live. So this tumor suppression occurs from turning off telomerase in your tissues. Because, in fact, most of us don't die of bone marrow failure. And so it limits lifespan to some extent, or in some people under some conditions, but overall it provides a tumor suppression mechanism, because reactivating telomerase is just another mutation that has to happen to those cancer cells. By removing telomerase, you have one more hurdle they have to pass to keep growing. But in the beginning, I mentioned that in cells without telomerase, human somatic cells, that get short telomeres, particularly if they're epithelial cells, they stop growing, but they sit there. And eventually, if they get induced to divide and have genomic instability, very infrequently, they'll generate a cell that survives and has activated telomerase through the genomic rearrangement. So in fact, short telomeres imposed by turning off telomerase are tumor promotion also. And so this balancing act, you can imagine, of how much telomere you should have to not restrict growth, to how much telomere you shouldn't have so that you limit growth, and how much telomere you should have so that you don't encourage cancer, there has to be a balance. So what would an ideal solution be if you think about this for a minute? The ideal solution, you don't have to be telomerase positive. You don't have to have active telomerase to keep growing. You just need long telomeres. So the ideal solution, you might imagine, would be to pulse telomerase on, to elongate telomeres, and then turn it off. And if you elongate telomeres to be able to meet the future demand for proliferation of that cell, and then turn it off, you get all expanding descendant cells, and all of them are more resistant to cancer. And lo and behold, that is what human development does. So telomere shortening in human somatic cells, but the much smaller number of cells in the germline and the many, many cells of early embryogenesis gain long telomeres. They reset each generation, unless you're compromised for telomerase genetically. And in that manner, tumor shortening can provide telomere suppression. So here's a graph of telomere length as a function of age, with lines kind of starting here. So maybe in this range is the human population genetic variation in your telomere lengths in your germline. And you can imagine that starting from either end of this, these germline cells know where to put their telomere. They put it at a certain length, let's say 10 kb. And then early on in fetal development, they turn telomerase off, and they can't maintain the telomeres anymore. So then, in further development, in infancy and childhood and adulthood, the demands for cell proliferation are counted. There's a lot of proliferation here, but here in adults, in fact, less. So in this range is a safe zone where telomeres can support somatic cell function and not contribute to tumors. And here what's just shown is in the germline where there's not proliferation, over time, because these cells are not dividing, they keep their telomere. And so these germline cells go on to make the next generation and start this process again. So in fact, in a multicellular organism, turning telomerase off in somatic cells is a mechanism of tumor suppression. And it's interesting to think about, then, this population variation, and also interesting to think about how this relates to medicine. Because you can imagine that if you're an adult over here, and you get chemotherapy, it wipes out your blood cells, and those hematopoietic stem cells have to launch into action again, right? You're asking them to repopulate an entire immune system with the telomere lengths of the immune system in an adult. Also, if you're thinking about a bone marrow transplant, for example, if you take bone marrow from an adult here, you transplant it, it's starting off with fewer variants. So there are applications of this towards therapy, for some screening for telomere length to inform towards some therapy. And I think in the future, through embryonic stem cells and through adult stem cells, knowing situations in which this telomere length can limit proliferation will be useful. And that's the last slide. For acknowledgements, I'd like to thank my many, many lab members and collaborators who make Cal Science happen. Yay! And have fun in the process. And in fact, I want to thank you greatly for your interest and attention, because it's a fantastic opportunity to just stand up there and tell you about what I love for 50 minutes. And for funding, early funding from the American Cancer Society, more consistent funding from the National Institutes of Health, and the continuing passion to find fountains of youth, existing ones such as my lab has tapped, and additional yet to be discovered.